Hi everyone. I just wanted to let you know that I'm recording this short introduction afterwards, just to make up for a small technical issue at the very beginning of the live session. So, welcome and thank you for being here. Today, or rather here, I will be talking about interpersonal synchrony. What it is and why it matters, particularly in the context of autism and caregiver-child diets. I'll walk you through key concepts, examples, and current research findings, and hopefully open some questions for future exploration. To begin, what exactly do we mean by interpersonal synchrony? Interpersonal synchrony, sometimes called interactional synchrony, refers to mutual dynamic adjustment of behaviors, emotions, biological responses between individuals during interaction. It is considered a key indicator of a dyadic functioning. It tells us not just about each individual, but about how the relationship as a system operates. So, synchrony is not about being identical. It's not simple simultaneity. It's about dynamic co-regulation and mutual attunement. So, now that we've defined interpersonal synchrony, let's make it more tangible. Synchrony is measured in many different ways. At the behavioral level, we observe it through things like coordinated movements, shared gaze, synchronized speech rhythms, and responding to a child's focus of attention. Biologically, synchrony can involve neuroalignment, when two brains activate in similar patterns, or physiological coupling, like heart rate rhythm sensing between parent and a child. We also see synchrony at the hormonal level, for example, oxytocin levels rising together during moments of positive social engagement, or cortisol patterns aligning in response to stress. These different forms give us multiple windows into how do people adapt, connect, and regulate together across behavior, body, and a brain. So the examples I just showed can actually be grouped in two main levels of analysis: behavioral and biological, each capturing a different layer of interpersonal synchrony. So at the behavioral level, we focused on observable actions, how bodies, faces, and voices align in both timing and meaning during interaction. So the behavioral synchrony is not the same as simple synchronization, which is more commonly seen in analysis of biological signals. Behavioral synchrony reflects something deeper, like temporal attunement, the ability of two partners to respond adaptively and contingently to one another.
Studying behavioral synchrony requires analyzing real interactions, and researchers typically use one of two main approaches to do this. So today we had an example of microanalytic coding, which involves coding specific behaviors like gaze shifts, gestures, vocal turns, frame by frame or second by second or even by event. And we also have global or macro level ratings. It involves making broader judgments about interaction quality over a longer stretch of time, focusing on aspects like emotional tone or mutual engagement. So this approach is more efficient, but it's less detailed. Okay, so let's look on the biological level. So biological synchrony can be measured across multiple systems: autonomic, endocrine, and neural. We track heart rate variability and respiratory patterns using ECG, monitor electrodermal activity through skin conductance sensors, and assess hormonal synchrony by measuring cortisol or oxytocin levels from saliva or plasma samples. Neurosynchrony, or brain-to-brain coupling, is typically captured with fMRS, EEG, while eye tracking systems allow us to study the synchrony of visual attention and focus. So together, these measures offer complementary insights into how physiological systems align dynamically during interaction. So now let me take a step back and ask why study interpersonal synchrony in autism. We know that about 1 in 100 children worldwide is diagnosed with autism according to World Health Organization. But autistic traits, the kinds of differences in social communication, sensory processing, or behavioral patterns that we associate with autism, actually exist across the general population. In that sense, autism isn't a separate category, but a point along a broader, multidimensional continuum of human variability. Autism spectrum condition represents a distinct profile at the higher end of that spectrum, not a single condition, but a diversity of strengths and challenges. Despite this, much of psychological research still treats typical development as a norm and deviations from it as its deficits. This creates a narrow view of what development should look like. In our project, we take a little different approach to that. We're not searching for what's missing, but what's different and what we can learn from these differences. Because at the end of the day, early interactions between mother and child, those micro moments of connection, are building blocks of later relationships. And if we want to support all children, we need models that reflect the full diversity of how relationships develop. Okay, how much do we really know? There are many challenges in studying synchrony. Far more than I can cover today, it's not the subject of this presentation, but if there's one core message I'd like you to take away from this slide, it's that most studies focus on parental behaviors, what they're doing, what they're saying, the gestures, while the child's active role is often overlooked. But synchrony is not something that happens on one side, it's dynamic, reciprocal process, a two-way street shaped by mutual adaptation of both partners. If we only study one participant in their interaction, we risk missing what synchrony truly is. And more broadly, when we look at studies of caregiver-child interaction, we see another issue. They often treat the caregiver-child relationship as a static unit without accounting for the individual characteristics each partner brings, like temperament, their cognitive style, or even autistic traits in parents themselves. So right now it's fair to say we know less than we know more. And that's not a failure. It's a reminder to approach this field with humility, curiosity, and a commitment to doing better science. So now I will walk you through the key findings, both between group comparisons and what we know about variability within autistic diets themselves. So, some studies show reduced synchrony in autistic diets. Autistic diets, I mean that there's the children with autism. These diets tend to spend less time and share positive emotional states and more often show mismatched emotions, like when the child is distressed but the parent stays positive. Parents may also use fewer synchronized gestures, especially in spontaneous and structured situations, which suggests that real-time coordination may be more difficult. Verbal synchrony is sometimes lower too. For example, parents may offer fewer comments that follow the child's focus of attention. But other studies show comparable synchrony between autistic and non-autistic diets, even in the everyday interactions and situations. What's really important and interesting is that higher synchrony, when it occurs, it has been linked to better long-term outcomes, such as language development or joint attention. And in some cases, caregivers, especially mothers, seem to adapt their behavior to maintain or even increase synchrony when a child engages more actively. This might reflect some kind of a compensatory strategy to support their interaction. So let's now turn what we know about biological synchrony, starting with physiological synchrony. We see some group differences, but also important nuances. For example, one study found that lower respiratory sinus arrhythmia synchrony in autistic diets compared to typically developing diets, but this synchrony increased when the interaction was of a higher quality and when the child had fewer internalizing symptoms. So again, the relational context really matters. Another study looked at cortisol responses during still phase paradigm. So children with autism show blunted stress response, and by that I mean that they didn't show initial stress response as typically developing children when mother was unavailable. But this happened only with mothers, and the interaction with their fathers was very similar to typically developing dyads. So this suggests possible maternal buffering, scaffolding effects, and emphasizes the need to include both parents in research. In terms of hormonal synchrony, Feldman found that children with autism start interaction with lower baseline oxytocin levels, but after interaction with a parent, both groups show a similar increase in oxytocin. So this speaks to shared biological potential for attunement. Moving to neural synchrony, results are also mixed. For example, autistic dyads did not show enhanced brain-to-brain coherence when interacting with their own parent, unlike typically developing dyads who did, in comparison to interaction with parent and a stranger. But in another study, children with autism showed increased frontal neural synchrony during cooperation tasks, although this was also linked to lower autism symptom severity. Other studies found no significant group differences, especially in more structured tasks or in early infancy. So overall, biological synchrony research reveals both similarities and differences, shaped not just by diagnosis, but the context, symptom profile, and relational dynamics. While many studies compare autistic and non-autistic dyads, it is important to recognize the diversity with autism themselves. First study showed that synchrony tends to be higher when both the parent is sensitive and the child is more responsive. So it's a co-created process. Second, contextual factors matter. For example, higher parental education has been linked to better synchrony, perhaps due to more researchers or knowledge about child development. Third, the parent's mindset plays a role. Parents who can tune into child's inner world and perspective tend to engage in more synchronous interactions. And finally, studies also highlight the role of fathers in the use of parentage, so that syncs on the way we talk to a child, and promoting engagement, especially in early infancy. So overall, synchrony in autism is heterogeneous. It's not just about diagnosis, it's shaped by individual traits, relationship, and a broader context of the interaction. So let's now look briefly what we know from longitudinal studies. One key finding comes from Kellerman. Dyads that showed higher synchrony at 12 months predicted better language skills at 36 months. And within the group of autistic children, more responsive parenting was linked to better cognitive outcomes. Another important contribution comes from Saint-Georges, who found that reduced social initiative and joint exploration were already visible in infants later diagnosed with autism, so even before their first birthday. These early differences were subtle, but they become more pronounced after first year. Similarly, in another research, researchers showed that infant siblings of autistic children often considered high risk, displayed weaker synchrony during infant-led play, suggesting that interactional differences can appear early, even before diagnosis. But we can also think of it as a broader autism phenotype. These children don't need to get the diagnosis. Maybe their behaviors are in the context of, I forgot the word, broader autism phenotype, yeah. So finally, it's just not about what we study, but also how we study it. A study by Kellerman showed that different methods of measuring synchrony may show our different findings. Global ratings picked up more early differences than macro-coded data, which is surprising. But this also suggests that some group-level differences might be more visible when we take a broader, holistic view of the interaction. So the findings we've seen so far reflect wide variability, but not just in behaviors, but in fit between partners. This invites us to move beyond individual explanation and consider a more relational lens. So the dialectical misattunement hypothesis offers an important shift into how we interpret social challenges in autism. It suggests that we often describe something as social deficits, that it might be actual relational mismatches. So from this perspective, synchrony isn't just something that autistic individual fails to achieve. It's something that emerges or breaks down in interaction, depending on how both our partners adapt to each other. This reframes our questions. Instead of asking what's missing, we start asking what's misaligned. and how we can maybe repair it together. And this hypothesis is not just theoretical. It's supported by studies which show higher synchrony in diagnostically matched pairs, like two autistic partners, than in mixed pairs with one autistic and one typically developing partner. So that suggests the relational compatibility, not diagnostic status alone or the ability of individual. So the compatibility may be key to successful coordination. So where are we now? The short answer is it's complicated. There's no single conclusion about how synchrony functions in autism. Findings are mixed, methods vary, and definitions are inconsistent. And as you could have noticed with the finding slides, I've color-coded the results to reflect the diversity. The red for reduced synchrony, the blue for comparable, and green for sometimes enhanced synchrony. So this variability isn't a noise, it's a signal for us. It tells us something that synchrony is dynamic, context-sensitive, and shaped by both partners and their environment. From what we know, we can cautiously suggest that synchrony in autism is likely different, not worse, not less, and these differences may reflect alternative relational patterns, not deficits. So to move forward, we need studies that look at both caregiver and child. Include mothers and fathers, go beyond diagnosis, and consider autistic traits dimensionally, even in parents. Use multimodal measures and better methods, and most importantly, follow development over time. Rather than looking for one answer, maybe we should ask better questions. How does synchrony develop? What supports it or hinders it? What does it mean for real relationship? The future lies in studying synchrony more carefully, more inclusively, and as a truly dyadic process. So after reviewing the evidence and reflecting on where the field stands, I'd like you to leave you with one final thought. Synchrony isn't a checklist of behaviors or numbers on a graph. It's about how two people meet each other in time, how they tune in, respond, and adapt. And I could put it in a metaphor and say that in autism, the tuning may sound different, but it's still music. So our job isn't to judge how typical the rhythm, the notes are, the music, but to understand what makes it work, and for that child and in that relationship. Oh, it's a variation. That's references, and thank you all for listening.
Thank you very much. So as you could, perfect, Julia. So as you could hear, there's more questions than answers. So it's good that we gathered here to try to examine synchrony further and try to answer so many questions. So we will leave the questions for the end because we have tight schedule, and then another presenter is waiting for us on Zoom. So I will try to let her in.